The hypothesis of this lab is that osmosis will occur in the beakers because the levels are not isotonic between the bag, like what's in the bag and what's in the beaker. You fill up 2 dialysis tubings one with a starch solution and one with a glucose solution. And the starch solution is bag A, and you put it in beaker A, which contains distilled water and glucose iodine, which gives it a yellow color. And then bag B contains a glucose solution, and you put it in beaker B that just has distilled water in it. And then you test the glucose of the distilled water in beaker B with a glucose indicator, and it should come up with a green because there shouldn't be any glucose in distilled water. And then you leave the bags overnight, and on the next day you measure the glucose levels in beaker B and do your observations. The next day you'll observe that the bag A is now like a bluish black because osmosis occurred and the glucose iodine and distilled water went into the bag and turned the starch color a blue. And then beaker B, you will measure another glucose strip. You'll measure the distilled water again, and you will see that the glucose strip turns a brownish green, and that indicates that glucose levels are present in the distilled water, which proves our hypothesis that osmosis occurred between the bag and the distilled water in the beaker.